Thank you for the kind introduction. Can you hear me? Delighted to have the opportunity to tell you a little bit about.
Could you get closer to the mic?
Is that better?
Yes.
Okay.
Yes.
Yes, that's better.
Could you lower the light a little bit? It's a little glaring.
Okay. All right. Yeah.
Sorry about that.
Tell me when it's comfortable.
Okay. That's better. Thank you. Yeah, delighted to have the opportunity to present EM-113, our autism program. It's a noncompetitive fast off-rate NMDA receptor antagonist for the treatment of autism in patients with elevated glutamate level in the anterior cingulate cortex as measured by proton magnetic resonance spectroscopy.
Is it working? Use the arrows.
That's better. Okay, that works. Thank you. So a quick thumbnail about the company, EuMentis. We're a clinical stage company developing neurocircuit specific therapeutics with initial focus on Tourette's and autism, both characterized by the neurocircuit imbalance leading to tics, cognitive deficit, and social deficit. And there are both programs, EM-113 and 221, are phase 2 ready, being teed up for first in patient toward the end of first quarter 2024. And as I mentioned, EM-113 is an noncompetitive fast off NMDA receptor antagonist. It's a reformulated memantine combined with a proprietary patient selection biomarker prospectively designed for autism patients with elevated glutamate level. It's a clinical validated target de-risked by control study, pilot study conducted by Joshi Gagan at Mass General Hospital, laying the mechanistic groundwork for our robust phase 2 clinical proof of concept study. This will be using a accelerated 505(b)(2) pathway. The second program is a best-in-class PDE10 inhibitor we licensed in from Mochida. It's well known that the blockade of that receptor, which is predominantly expressed in the striatum in the central compartment, recapitulates the D2 inhibition without the metabolic, cardiometabolic side effect compared to the typical neuroleptic agents, the second generation antipsychotics being used for this particular indication, such as risperidone and aripiprazole. There's also an ongoing interest in developing a second generation better memantine that specifically targets extrasynaptic NMDA receptor that are hypothesized to be involved in the excitotoxicity of the glutamate dysregulation. The fundamental challenge, I think it's a recurring theme throughout this conference, is that the heterogeneity and the genetic variance of ASD perhaps has contributed to the high attrition rate in the clinical study of targeted therapy. Subset of population may well respond to a targeted therapy, does not necessarily translate to the broader response in the intended to treat population of patients who may have dysregulation of other pathways. So there's a significant unmet need to develop biomarkers that are predictive, allow for patient selection, who may, in a predictive manner, respond to the targeted therapy. Recognizing this particular challenge and the critical gap, in the last couple of decades, Josh Gargan at MGH have interrogated the glutamate dysregulation in a specific region of the brain, anterior cingulate cortex, and have found that there's elevation of the glutamate level in the ASD patients compared to the normal control. Expanding that finding, recently he has done a pilot study demonstrating that memantine benefits the patients with the elevated glutamate level in that subpopulation. And that was an all-comer study. To build on that, in collaboration with Dr. Gargan, we have licensed his biomarker, proprietary, and designed a robust phase 2 study using a differentiated formulation of memantine with a different dosing regimen combined with this proprietary biomarker as a diagnostic tool to stratify patient population. And we're currently in discussion with the FDA to iron out a couple of regulatory questions to be able to initiate that study. Converging experimental evidence provides mechanistic support for a strong link between glutamate dysregulation and autism spectrum disorder. Through the NMDA receptor, glutamate is involved in, plays a critical role in maintaining synaptic plasticity, cognition, as well as social interaction. And under the physiological condition, while under sustained excessive excitation, the signaling, the excessive excitation leads to neuronal death, and predominantly through the extrasynaptic receptor, as illustrated elegantly by earlier work coming out of Stuart Lipton's lab down at the Scripps. And the excessive excitation of the NMD receptor is also thought to create that disequilibrium or imbalance between excitatory and inhibitory inputs. Here is a slide summarizing some of the key compounds that have been thoroughly characterized over the decades. And NMD receptor is considered a bit of a target du jour going back to the 90s. Every large pharma company had a program. And I think the major breakthrough really came out of Stuart's observation that high affinity, slow off rate drugs are not well tolerated. And as you can see from the blockade of the current by MK801, it's a dose-dependent manner, but almost in a insurmountable fashion, that the drug doesn't come off, cause sustained, more protracted inhibition of the current, and perhaps explain the lack of safety tolerability profile of this drug in preclinically, and causing hyperlocomotive side effect. On the other extreme of the spectrum is memantine, which has a unique profile in that it causes dose-dependent inhibition, but it has a very rapid off rate, so that it doesn't interfere with the physiological function of the glutamate transmission, while allowing for inhibition of the extrasynaptic receptor that are involved in the excitotoxicity component under pathophysiological condition. As he illustrated in the hippocampal neuron prep, the synaptic excitatory postsynaptic current can be inhibited or inhibited to a very limited extent under physiological condition using a therapeutic concentration of memantine. As you can tell, that the inhibition is fairly minimal, while the inhibition under pathophysiological condition, where there's large concentration in MDA, to recapitulate that pathophysiological condition, you get a very significant, more exaggerated inhibition, leading to a selectivity of favoring extrasynaptic receptor and relatively sparing toward the synaptic transmission that are physiological. There's a significant interest in developing a better memantine, and we have a couple of compounds that are under profiling as potential development candidate. 036 is one discovered by Stuart Lipton. Again, this is a dual pharmacology compound. Has a NO donor component that perhaps provides a more tonic inhibition of the receptor against the, again, with an improved extrasynaptic receptor selectivity. All three compounds have a rapid off rate, similar to memantine. Now, Galgana at MGH have demonstrated that have detected in the ASD patients an elevated glutamate level in the using the proton magnetic resonance spectroscopy, able to isolate the glutamate signal, which is around the 2.3 ppm, away from glutamine and GABA, able to quantify and demonstrate that there is a significant elevation of that glutamine in the region of anterior cingulate cortex, which is involved in social information processing. And that data is fairly consistent with the prior finding by other groups that the dorsal ACC was hyperactivated in patients with ASD versus the neurotypical subjects in normal control. And that seems to suggest the additional effort is required to process and execute facial identification task. And the author, Gabriel Richter, was also able to demonstrate a degree of activation in ACC is inversely correlated with social impairment severity, which is somewhat counterintuitive, but may reflect a compensatory mechanism in those patients. Taking that finding further, with the permission of Dr. Galgan, I'd like to describe some of the top-line findings from the his recent pilot study, which is under review for publication. And it's an all-comer study in the pediatric population of age 8 to 17, intellectually capable, and with social impairment. It's a relatively small study, 40 subjects, and 12-week of one-to-one randomization between twice daily 10 milligram dose versus placebo, and magnetic resonance spectroscopy of glutamate level is one of the, it's not an inclusion criteria, selection criteria, but it's used to study the temporal change of the glutamate level at screening versus end of the study, but also how does that correlate with, line up with efficacy. The study was positive based on a responder definition of 25% reduction in social responsive scale, change from baseline 25%, and clinical global impression of less than 2. Fairly significant improvement was seen in the treatment group, 56% versus the 21%. And if you dichotomize the patients based on high glutamate versus low glutamate sample, it turns out that the main driver of the efficacy was really the high glutamate subjects. So it's entirely, in fact, driven by that subpopulation. And taking another data cut and looking at the temporal profile of the glutamate level at screening versus end of the study, the consistent pattern illustrates that for the high glutamate sample, you have a clear reduction in glutamate level compared to time 0, and very little change for the normal glutamate sample. And that seemed to be congruent with the clinical outcome readout. And this really provides for the first time, a rational selection of a biomarker that we hope that's through our next study so before I jump on to the next slide, this is a slide really summarize what was previously done by Forest Lab. And as you may well know, that memantine is being investigated in that same indication, same population previously by Forest Lab. And it was a negative study, relatively large. But looking back, retrospectively, there are a number of limitations associated with that study that contributed to the failure. One is it was a relatively low dose, significantly lower than what was considered efficacious for the approved indication, the dementia of associated with a moderate to severe Alzheimer's type. And that low dose is likely contributed to low receptor occupancy, subtherapeutic exposure, no focus on intellectually capable subgroup, allowed for enrollment of patients with significant intellectual disability. Inclusion criteria for the social responsiveness scale was too low. And there was no biomarker. And not surprisingly, whatever they have seen in earlier study with smaller sample size did not necessarily translate into the all-comer broader population. With all that in mind, EuMentis has embarked on a large phase 2 study using this biomarker precision medicine approach. The objective is to test the memantine specifically in the high glutamate population to determine hopefully to demonstrate the predictability of that biomarker in predicting patients who are likely to respond. The magnetic resonance spectroscopy will be an integral part in the patient selection, stratification. So the key inclusion criteria is ASD diagnosis, IQ requirements over 85, and social responsiveness scale over 85. And this will be a 12-week study, randomized 1-to-1 against placebo. The active will be the patients titrated to up to maximum of 24 milligram. The primary outcome will be the social responsiveness scale. We'll be looking at also categorical responder analysis as exploratory. And we're still in final stage of discussion with FDA about using the high dose group in the adolescent population, hoping that we'll be able to include the adolescent post-pubertal population in this study. Increased chance of success, similar to the Joshi study. And so historically that's been concern about Oni's lesion with this class of compound, especially for high affinity drugs. Nonetheless, that's been an ongoing discussion to convince the agency that the 24 milligram, the lower dose, would be safe for the adolescent population. With that, well, thank you for your attention, and we have Randall, who's been the main driver of all those programs, who's a card-carrying neurologist, will be able to answer the questions that I'm sure will be beyond my ability to cover. Thank you for your attention. It's a pleasure to be with you at the conference.
Thank you, Dr. Zhang. We have a few questions here from Evdo.
Hi, thank you. Nice to see Mementing coming back. At the first time around when we were doing the Mementing studies, the reason we were capping at 12 years is because FDA was worried about apoptosis peripubertally in the brain based on the animal model. I see the age going up. Do we have a resolution on that concern? They had actually shut down previous attempts to go above 12.
We'll let Randall take that. We can't, his audio is not coming through.
Priya? Are you with?
Increase the volume here.
We can hear you here, but I don't know why. No, it's too. Yeah.
Oh, you need to like maximize volume with it? Write your ringtone. Oh, yes. Does this thing have a headphone jack? go.
Yeah?
There we go.
All right, we can hear you now.
Okay. Well, unfortunately, I couldn't hear you. I wasn't able to hear the question. There was too much echo in the room or something. Can you repeat it for me, Xiaoming? Because I can hear you fine.
The question was about the clinical hold on the Forest Lab program because of the Odin's lesion finding and whether we're getting closer to resolution with FDA.
That is a great question. So can I ask, is the person there, was the person there actually involved with the forest trial? Which several years ago.
I think you may want to say that back because he can't hear you. You can communicate her response to Dr. Marshall.
Where, do I use the microphone or?
Yeah, I don't think. Hi, Randall. It's Evdokia. So at the time, there was the forest study and there was an investigator-initiated study by Autism Speaks. They both got shut down for anybody above. Well, we got shut down for a good 2 years, and then it got released back for only kids under 12. So they had made the decision at the time that they would not allow peripubertal kids in the study.
Did you hear?
Yeah. Randall, can you hear me?
I can hear you only, nobody else.
Okay, sorry about that.
Okay, just
So-
repeat the question for me.
Yeah, so let me just repeat the question. There was a investigator-sponsored trial she was involved in. At the same time, the Forest Lab program was put on hold. So
Okay, so that's how she's aware of the controversy. Yeah, well, so the letter from the FDA to Forest is in the public domain. The FDA required a 10-fold margin of safety below the NOEL based on some probably controversial interpretations of the toxicology data in the juvenile rodent study. And so the position, which for Forest, and I think has continued for probably all of the NMDA antagonists in front of the agency, is that it would be, the only lesions are neurodegenerative lesions in the rat brain. And that's considered unmonitorable and irreversible based on the rat data from a long time ago, actually. And so they have taken a very, very cautious stance. It really comes down to whether you, and I'd like to say also that those lesions have not been found in multiple other species. And so there's some thought that this is due specifically to elevated metabolism in the rodent brain and specific parts of the brain where the only lesions occur. There have been studies in autopsy studies in Alzheimer's patients who've been on memantine for years. There was no evidence of anything resembling an only lesion. They don't show up in primate studies. But you know, we understand wanting to show an abundance of caution with children, but we think that in autism that the risk benefit is worth being a little more flexible on the margin of safety. And we're not asking to completely ignore the margin of safety, but we're asking for a little more flexibility. You know, what about 6-fold margin of safety? What about 7-fold margin of safety? You know, and just a shift in a few from 10 to 7, for example, lets you get much closer to the dose that Dr. Joshi found was effective in this subpopulation. So we continue to have this discussion, and we're hopeful. We'll be going back to the agency for a Type D meeting in a couple of 3 weeks, and we hope to have a face-to-face dialogue. I'm sure any other company that's been developing an NMDA antagonist has had a very similar conversation.
Very,
All right.
very good question, and I think you picked on the fact that we're taking a stepwise approach. And staying below the exposure margin that's gonna be insisted by the agency. Once we've established safety and efficacy in the adult population, the hope is that we'll be able to justify going into the adolescent, lower age groups.
Hello, I was wondering if the patients that had the highest glutamate peak by MR Spect in the ACC, were there any clinical correlates to them? For example, were they more stereotypic, or did they have more SIB, for example?
Randall, did you hear the question? You want me to repeat it?
No, just repeat it for me.
Okay, so the question is, from Joshi's study, whether the high glutamate population has clinical correlates in terms of severity.
No. The short answer is no. And you know, I'm a clinician myself, and I worked in academia at Columbia for many years, and we were always searching for a clinical profile or syndrome that could correlate with the biology, and we never succeeded. There have been many sophisticated attempts to do that, and similarly, there was no phenomenological phenotype pattern that went along with the high glutamate. It's not surprising, because autism is a multifactorial disease. It's a very complicated manifestation, and we suspect that, like probably all of the major psychiatric conditions, there are multiple biological pathways to the same phenotype. And so we're convinced that the right way to go is to focus on a biological biomarker rather than continuing to find a phenotype. And, you know, the other issues that with these syndromal extractions from the symptom counts, even if you identify one, they tend to be very weakly predictive of things like drug response and clinical outcome and clinical course. So we are really excited about this finding. When I met Dr. Joshi at MGH, he's been incredibly generous with his data, and it has really allowed us to move forward with a plan to enrich our sample with high glutamate patients based on his pilot work. And we're, like Xiaoming said, we're starting Q1 of next year. Maybe there are some PIs in the audience who have already agreed to be a site, but if you're interested and you haven't heard from us yet, then please let me know, or let Xiaoming know after the meeting.
And along those same lines, might there be an EEG correlate to that increased glutamatergic tone, for example, excess beta activity in the ACC or something of that nature?
Are there any direct EEG evidence in terms of correlation with, are you referring to the glutamate?
Oh, you mean in this study? It wasn't measured.
There were no EEG in that study.
Yeah, so this focus on the glutamate level in the ACC, there are only a few studies, and I don't think that an in-depth characterization using multiple other biomarkers that show up on EEG, like gamma, gamma is altered with memantine, for example, in humans as well as animals. It just hasn't been fully characterized to my knowledge. At least I haven't seen it published.
All right, thank you. Actually, I have a follow-up question on that as well. You mentioned that there were no clinical correlates of the biomarker that Dr. Joshi has for glutamate levels. If that's the case, is there a reason to limit the study to those that you said were intellectually capable versus not? Is that more because of ease of implementing the assessments or some other reason?
If you could maybe repeat it on this mic.
That's maybe easier than translating it for me, yeah.
just to make sure, I think the question was lost in translation.
Dr. Marshall, this is Sarika here.
Yes, hello.
Hi. So I had a follow-up question on the previous one about the clinical correlates, right? You mentioned Dr. Joshi's biomarker for glutamate levels. So if there were really no clinical correlates, is there a reason for why the study enrollment is limited to those who are deemed to be intellectually capable versus not?
Yeah, that's a great question. So this is a special area of interest for Dr. Joshi in his clinic. It has been for, I think, quite a few years. The therapeutic approach to patients who have who are intellectually capable, meaning they're conversant, and they often have normal or even high IQs, but they're profoundly socially impaired. The therapeutic approach can be quite different from autistic patients with much more severe functional impairment, patients who have little or no language, patients who have profound behavioral abnormalities. And so he decided to first focus on that population clinically. And then I think his work carried into the ACC based on some of the experimental data. In the fMRI studies, focusing on the ACC and its relationship to social information processing. And then from there, I think he just had the idea that if glutamate was elevated, then an NMDA antagonist might have therapeutic benefit, and convince the NIMH to fund the work. So the other thing to know is that the scale itself, the social responsiveness scale, it starts to lose validity as patients become more generally impaired and certainly impaired in their communication skills. And like any observational or self-report or family report measure, it has a range within which it is valid.
All right. Thank you, Dr. Zhang.
Sure.
And now we're ready to move on to our next speaker here. Dr. Xiaoming here. Oh, sorry. So it is a great pleasure to welcome again our friends of the years. Dr. Xiaoming is the director of the Autism Research Program at Autism University. His research focuses on the medical cause of autism and how the treatment symptoms, including the areas of neurosis, oxidative stress, gut bacteria, antibiotics.
Soundtrack.
Not nice one. Look it on the other one. Like a connection. The research scientists in the biology center of to microbiome organization. He got closely Dr. Brown and Dr. Adams and what microbiome and focused on developing